We can't express how grateful we are to have all of you here. We can't express how grateful we are to have all of you here with us today and providing your huge support, kind cooperation for this consortium biotech workshop from around the world. Big thanks from all of us. As discussed earlier in the previous workshops, the main motto of this workshop series are based on the statement given by our father of nation, Sri Mahatma Gandhi ji: live as you were to die tomorrow, learn as you were to live forever. Hence, these workshops were designed for the following purposes. Purpose 1, to avoid the error at the time of designing of our experiments and during the bench work as well. Second purpose of this consortium biotech workshop is School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya Bhopal is having the almost all facilities to pursue the research work at the various aspects of molecular medicine and also environmental biotechnology. We are ready to extend our such facilities to the interested ones. These workshops, as we discussed earlier also, Workshop 1, how to start the lab work under aseptic conditions, which we have already completed on 1st and 2nd of June 2020. Workshop 2, how microbes are fascinating, that was done on 7th and 8th of June 2020. Workshop 3, we all are assembled here for the third workshop, that is how to study the beauty of nucleic acids instead of DNA. That is DNA and RNA both. That is on today and tomorrow on 14th and 15th of June 2020. And the upcoming workshops are Workshop 4, how the gene is expressed in the form of protein. That is to be held on 21st and 22nd of June 2020. Workshop 5, how computer helps in the designing of biological data. That is on 28th and 29th of June 2020. Now, the time for the agaz of the today's workshop. This today's workshop is divided into 5 sessions. For today, we are going to deal with the 2 sessions. In session 1, extraction and isolation of DNA from animal, extraction and isolation of DNA and RNA from human tissues. These we will discuss in the first session. In the second session, extraction and isolation of DNA from plant. Tomorrow, 2 sessions, that is third and fourth. During the third session, we will discuss the extraction and isolation of RNA from viruses, particularly the COVID-19, which type of test we are going to perform, whether it is positive COVID-19 or coronavirus positive or negative, COVID-19 positive or negative, we will discuss under this. Extraction and isolation of DNA from bacteria, that is plasmid isolation, genomic DNA isolation. Followed by purification and quantification of DNA and RNA, gel electrophoresis of DNA and RNA, that is preparation of the gel, loading of the samples, running of the gel, and visualization of the bands on the gel. These all we will discuss in the third and fourth session tomorrow. In the fifth session, that is followed by feedback session. Whatever we will discuss today and tomorrow on the basis of the dear participants' valuable feedback. Now, we are just introducing Dr. Suresh Kumar Jatwa. He is going to present on the basics of DNA extraction and isolation from animal tissues. Dr. Suresh Kumar Jatwa is Assistant Professor, School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, University of Technology of Government of Madhya Pradesh, accredited with grade A by NIC, Bhopal. Dr. Suresh Kumar Jatwa.
Thank you very much, ma'am. My slide is visible, ma'am?
Yeah, please carry on.
Good evening, all of you. So this is the first session of workshop third, that is how to study the beauty of nucleic acids. So today we will discuss about the extraction and isolation of DNA from animals. So in today's session, the topics to be discussed is about DNA, properties of DNA, need of DNA isolation, DNA isolation from tissue, DNA isolation from blood, and applications. So as you know, DNA isolation is a process of purification of DNA from nucleus because DNA is found in the nucleus of the cell. And it is the process of the combination of physical and chemical methods. So it is obtained to obtain DNA in relatively purified form, which can be used for further investigations like polymerase chain reactions, signal sequencing, diagnosis, etc. etc. The first DNA isolation of DNA was done in 1869 by Friedrich Mischer. So always remember the methods used to isolate DNA always dependent on the source from where you are getting the sample, age and size of the sample. The most common purpose of DNA isolation is it is aimed to separate DNA present in the nucleus of the cell from other cellular components. And isolation of DNA is needed for genetic analysis, which is used for scientific, medical, or maybe forensic purposes. DNA is having number of applications such as in production of DNA into cell and animal or plant or for diagnostic purposes. So there may be various sources from where you can get the samples for the isolation of DNA. So DNA can be isolated from any living or dead organism. Sample may be or source may be whole blood, hair, sperm, bones, nails, tissues, blood stains, saliva, buccal swabs, epithelial cells, urine, etc. Stored, sometimes stored sample also we can use that come from the archive tissue samples, frozen blood or tissue, exempted bones or tissues, and ancient human, animals, etc. So before isolating the DNA or going for isolation of the DNA, first of all, we have to understand the basics of the DNA, like what is DNA, what are the building blocks of DNA, or where DNA is located, and chemical properties of DNA. So here you can see these are the Watson and Crick, the picture. So in 1953, when they have given the double helix structure of DNA. So DNA is deoxyribonucleic acid, and it is a polymer, or we can say chain of nucleotides. And what is nucleotides? Nucleotides is made up of, they are made up with a nitrogenous base, a sugar that is deoxyribose, and with phosphate. These nucleotides interconnected together to form a chain which is known as the strand of DNA. It is only the molecule that encodes the genetic instructions used in the development and functioning of all knowing living organisms. That's why it is known as the blueprint of life or hereditary material of an organism. So when we discuss about the chemical structure of DNA, as we discussed in my previous slide, the building block of DNA is nucleotide, and nucleotide is composed with a nitrogenous base, with a sugar that is deoxyribose, and with phosphate. So the nitrogen bases may be of two types, that is purines, means adenine and guanine, and pyrimidines like cytosine and thymine. So any of the four nitrogen base are connected with the sugar, further attached with the backbone like phosphate, it makes nucleotide. Here we can see these are the two strands of DNA, parallel strands. We can see here these are anti-parallel. This 5 prime and 2 3 prime strand, and this is 3 prime and 2 5 prime strand, in which we can see this is phosphate, sugar, and nitrogen base. Here it is, in case it is adenine, and adenine always form attached with the complementary base, that is thymine, and guanine with cytosine. So here we can see this is the chemical structure of an DNA or nucleotide. So this is the nucleotide component is nitrogenous base, maybe adenine, thymine, guanine, or cytosine, and it is interconnected or connected by an glycosidic bond here between the first carbon of the sugar, that is deoxyribose sugar, and nitrogenous base, and this further attached with the fifth carbon of the sugar with phosphodiester bond. These three molecules, phosphate, sugar, and base, commonly known as a nucleotide. So, as we know, DNA is a double helix model given by Watson and Crick in 1953. So, it is a question how double helix structure is maintained. So, DNA is made up of two anti-parallel strands, as we discussed in earlier slide. That is, the first strand is 5 prime and 2 3 prime and another one is just anti-parallel, that is 3 prime and 2 5 prime. So, these always combined with the complementary
A
base.
lot of people are here.
Please mute your microphones, please. It is my humble request, please. So this is, as we can see in this picture, this is the one strand and this is second strand. And these are anti-parallel, bonded with the hydrogen bond between the adjacent or complementary base pairs, like thymine attached with the adenine and guanine attached with the cytosine. So like this, they form a hydrogen bond in between the complementary base pairs, like this: adenine, thymine, guanine, and cytosine. So hydrogen bond hold adjacent like this and complementary base pair together to maintain the double helix structure of the DNA. And this complementary base pairing always ensures that mismatch may not made during the replication of DNA or DNA transcription, because always these must be always in complementary form, like guanine to cytosine and thymine to adenine. And sugar phosphate here, it is hydrophilic in nature, so it is positioned just outside the structure to stabilize the double helical structure of the DNA. And here we can see the nitrogen base just positioned the inner side of the DNA or groove, like this. We can see here or we can see here. So these are nitrogen base. So these are very reactive. So they are positioned just inner side of the structure. Another question is where DNA is located. So DNA is located in the cell. It is primary thing, and cell is composed with these so many type of cell organelles. Here you can see so many type of cell organelles, and one of these organelle is nucleus. This is the nucleus. So in the nucleus, chromosomes are found, and the chromosomes having DNA. We can see clearly with this picture. Cell, this is cell organelles, nucleus. In nucleus, these are the chromosomes, and chromosome contains DNA, or chromosomes made up of DNA, which contains genes. So to study the DNA or study the genetic material or any type of gene function or any type of diagnosis, so there are so many applications of DNA isolated. So it is very important step how to take this DNA out of the cell. As we know, this is a particular cell, and cell nucleus contains chromosomes, chromosomes having genes, and genes are the functioning unit of the DNA, which is responsible for a particular protein, or maybe we can say particular trait. So how can we identify? So it could be identify whether you are able to isolate this particular DNA, and from DNA this particular gene from the cell. So here we will discuss about the isolation of DNA from animal cell. So these all practicals we routinely perform in our laboratory, that is School of Biotechnology at the PV Bhopal. So the procedure of isolation of DNA basically consists these four major steps. The first one is the preparation of cell lysate or preparation of cell extract, and second one is purification of DNA from cell lysate or cell extract, and constitution of DNA samples, measurement of purity or DNA concentration. So all these four steps we can understand with this pictorial presentation or representation. So this is the sample, this particular, so having your sample, maybe cell or tissue. So first step is sample preparation. It means you are going for the cell lysis or preparation of cell extract. Then the cell extract may contain some cell debris, proteins, RNA, but your need is only, your aim is to isolate only DNA, but it contains so many other things also, or cell organelles or cell components. So the removal of these particular things is necessary. So for this you are removing this particular things, like you are removing protein with the help of proteinase, or you are removing RNA with the help of RNase. Then now you are precipitating your DNA, then DNA rehydration with the help of opponent. Then other this with the help of column, this DNA binding, and you go for washing twice or thrice. Then you can elute your DNA in the pure form. Please mute your mic, please. It is my humble request. So the first step of DNA isolation from tissue or cell, that is the preparation of cell lysate or cell extract. So to extract the DNA from tissue or cell that is of your own interest or your need, so the first step is the cell have to be separated and the cell membrane have to be disrupted just to release the DNA from the nucleus. So the lysis buffer contains EDTA and SDS, helps in carrying out this process, like EDTA. So EDTA is ethylene diamine tetraacetate, which removes Mg2 plus ions that are essential for preserving the overall structure of the cell membrane. And this SDS, that is sodium dodecyl sulfate, which disrupts the cell membrane by removing the lipids of the cell membrane. By this we can rupture the cell membrane with the help of this, which contains DDTA and SDS in the lysis buffer. Once you have lysed the cells like this, your final step is the precipitation of cell lysate and removal of the any type of insoluble cell debris or contaminants. So like with this picture, this is the cell in lysis, and go for centrifugation. After centrifugation you will get the pellet, junk pellet, and some supernatant contains your material of interest. So just you keep and just you centrifuge again and wash it, then you will get your DNA in the second tube. Then purification of DNA from cell extract. So as I said, in addition to DNA, we have made cell extract. First step in cell preparation of cell extract. This cell extract, addition of the DNA, also contains significant quantity of detergent, proteins, RNA, salt, or reagent used during the cell lysis. So there are a variety of procedures that can be used to remove all these contaminants, or we can say the cell debris or the cellular components from the DNA to living DNA in the pure form. So most commonly used procedures are ethanol precipitation, phenol chloroform extraction, and mini column purification. So ethanol precipitation is the first one. So here in tube you can see the ethanol precipitation is carried out usually by ice cold ethanol or isopropanol, because DNA is insoluble in alcohol, so it will aggregate together. And I'll go for centrifugation. We will get a pellet at the bottom of the tube here. That is your DNA. So precipitation of DNA is improved, also can be improved by increasing of ionic strength using by adding sodium acetate. In this pictorial diagram, you can see also this is in nucleic acid solution, and alcohol, adding alcohol, precipitation nucleic acid, and centrifuge. Then all these remaining things you discarded and then the supernatant, and this pellet contains the nucleic acid of your own interest. Then you can resuspend it and isolate and separately in the form of this pure nucleic acid. And phenol chloroform extraction, it is a method also in which phenol denatures proteins in the sample, and after centrifugation, when you this is cell extract, that is the first step of DNA isolation. Then when you mix the phenol well in this cell extract, then it will separate the layers by centrifugation. Here you can see this is the aqueous phase containing DNA and RNA of, or we can say nucleic acid, and this is the interface by containing proteins, and this is phenol. So by taking very carefully this supernatant, you will able to get your nucleic acid, and by treating this particular this aqueous phase with the RNAs, then the RNA will degrade, and now you are able to isolate only DNA from this aqueous layer. And another one is mini column purification. It is based on the adsorption. So when you are having this sample prepared or cell extract or cell lysate, when you go for DNA binding, we pour all these sample into the column, and column having solid phase, that is made up of silica or another one. So by this passing the sample from this, your DNA will stuck there, bind this particular phase, and then you wash it twice or thrice. Then you can elute your pure DNA attached with this particular phase in given quantity, maybe 100 microliter or 150 microliter or 50 microliter as per your protocol. Then concentration of DNA samples. So in the presence of salt at a temperature of minus 20 degrees Celsius or less, absolute ethanol will efficiently precipitate polymeric nucleic acid. Here you can see this is cells or cell lysate using detergent that disrupt the plasma membrane or other things. Then this cell contained or treated with the protease just to destroy the proteins from the sample, and RNA just to destroy the RNA from the sample, so you can get only DNA. Then this cell debris is pelleted in the centrifuge. Then the supernatant liquid only containing the DNA is transferred to a new one tube, and this tube contains only your DNA, isolated DNA. This isolated DNA can be further pulled out with the help of this glass rod or maybe diluted them. The another example, that is the DNA isolation from blood. So as we discussed earlier, so this is common phases or these steps, that is first the preparation of the cell lysate or acid of the cell or cell disruption. Second one is the phase separation. Third one is clearing of proteins and clearing of RNA, then DNA precipitation from the blood. So being blood DNA isolation, the lysis can be done with the help of two methods, that is two-step method, and another one is one-step method. In two-step method, because blood contains RBCs and WBCs both, so first we go for RBC lysis and second we go for WBC lysis, so that the DNA can is from the nuclei of the WBCs. Then removal of all the contaminants or cell debris from this lysate, DNA can be recovered in the form of pellet like this. And while in one step method, WBCs and RBCs both lies in one step. Then nuclei and mitochondria isolated from the LUC by centrifugation. Then all this cell from the cell extract, all the contaminants or cell debris will be removed from this cell extract, and DNA can be recovered with the help of by following centrifugation in the form of pellet at the bottom of the tube like this. So second is the phase separation. So here you can see this is a tube, microcentrifuge tube containing blood or sample. So there are three phases. You can see this is aqueous phase. Second one is this is interphase containing DNA, and this contains RNA. And this is the organic phase, contains membrane lipids, proteins, or maybe polysaccharide, another type of cell debris. So in addition to the chloroform isoamyl and raised to 24 raised to 1, or phenol chloroform isoamyl alcohol and raised to 25 raised to 24 raised to 1, following centrifugation, all organic solvents, just I said, the membrane lipids, proteins, polysaccharide, or maybe some cell debris will keep trapped the bottom here. So by eluting this interface very carefully, by removing this aqueous phase, so removal of RNA took place, and now very carefully you take this interface which contains the DNA of your own interest, then you transfer it into the separate tube. So this will be your DNA. So what is the role of phenol? So here you can see this contains DNA, this DNA plus proteins. So proteins in aqueous solution. This is, so proteins are polar residues. Guys, this is located just outside of the simple, and when you are adding phenol, so phenol added and mixed vigorously. So while doing this protein are the less polar residues, just flip out. So just flip to outside, and by flipping outside, this lesser polar proteins become this side, and DNA polar comes outer side like this. So this is the role of phenol. While DNA is negatively charged molecules, so therefore soluble in polar solvent like water. So whereas proteins have both hydrophilic and hydrophobic side chain, ammonia and the amino acids, and in cell normally DNA remains dissolved in polar phase, and the proteins tend to fold in such a way that their hydrophilic or polar goes face outwards in this way in the solvent, and hydrophobic towards the inside. So on adding phenol, when you are adding phenol, protein flips. Since phenol is less polar than the water, so flip like this. So this protein becomes this side, and the DNA polar like this side. Then role of phenol and isoamyl alcohol. So we can say chloroform is significantly denser than the water. So adding it to the organic phase increases the overall density of that phase, helping it to prevent phase inversions. And isoamyl alcohol act as an anti-foaming agent in the sample. Chloroform, when added next to the phenol chloroform treatment, helps in extracting phenol from the aqueous solution. So this is the final step where you got all these things in the cell extract. So by clearing out proteins, because this DNA, this cell extract contains apart from DNA some proteins, RNA, or maybe some cell debris or cell contaminants or cell organelles. So by clearing out the proteins, RNA and DNA precipitation, you can get the pure form of DNA. So proteins can be eliminated by adding proteases, and RNA can be eliminated by adding RNAs. This particular treatment or step increases the purity of isolated DNA, and DNA precipitates with alcohol, usually pure and pure ethanol or isopropanol, because DNA is non-soluble in alcohol. So it will form aggregate and precipitate at the bottom of the tube like this after centrifugation. So by washing with the 7% ethanol, you dry it by opening the lid of the tube for 15 to 20 minutes or 30 minutes maybe at room temperature. Then you can dissolve this pellet in a given quantity like 50 microliter, 100 or maybe 150 as per the protocol. Then you will get the pure form of DNA. So these are the some of the applications of selected DNA. So in the applications, we can see scientists may use this DNA and like introduction of DNA into the cell or animal or maybe in plants. It is also used for the diagnostic purposes, and in medicine, the later application is most common in medicine also. It is also used for forensic science purpose because forensic science needs to recover DNA for identification of individuals, like we can say for the identification of rapists or for petty thieves or accident or war victims, etc. It is also used for the paternity determination and plant or animal identification as well. So these are the some of the applications of DNA isolation or isolated DNA. So this isolated DNA also requires to check whether it is in the pure form or not. So the next step or is after isolation is DNA purification or to check DNA purity and the quantification. So this particular section will be covered by in the next session by Dr. Mrityunjay. And thanks for your listening or kind questions. So now I would like to invite Riddhi Tiwari, who is a doctoral fellow at the School of Biotechnology, Rajiv Gandhi Pradyogiki Vishwavidyalaya, to discuss about the isolation and extraction of nucleic acids, means DNA and RNA both, from human. So, Riddhi Tiwari, please.
Thank you so much, Dr. Suresh, for such a wonderful presentation and inviting me to continue with this current session of DNA isolation of workshop 3 of Consortium Biotech workshop. So in continuation of this workshop, how to study the beauty of nucleic acids of Consortium Biotech workshop, I'm going to talk something about extraction and isolation of human DNA and RNA. So for this, let's have some glimpse of organization of human genome. Human genome means a total genetic information, that means total DNA content present in human cells. The human genome consists of two types of genomes. One is a nuclear genome, another is mitochondrial genome. I would request anybody who is opened his, please mute your microphones. It is causing some disturbance. Okay. So 99.99% of the total genetic information in the human cells comprises of nuclear genome, and the mitochondrial genome is the remaining of 0.0005%. What is a nuclear genome? Nuclear genome is a chain of DNA organized into chromosomes. In humans, there are 3 into 10 to the power 9 base pairs packed into 23 chromosomes, which means when the double the number of chromosomes, that is 2n is 46. The size of the human chromosomes ranges between 50 to 250 megabase pairs, and each chromosome is a single long molecule of DNA. What is a DNA? DNA is the basic unit of information present in a gene. It comprises of 4 nucleotides: adenine, thymine, guanine, and cytosine. The synthesis of DNA primarily occurs in 5' to 3' direction through the formation of phosphodiester bonds between deoxyribose and phosphate, which gives rise to the sugar phosphate backbone. DNA is formed in the double helix because of the hydrogen bonding between complementary bases. Specific sequences of bases give rise to protein structure and genetic inheritance. As per the Human Genome Project, it has been found out that we all have near about 30,000 genes in our body, in our cells. So what is a nucleic acid extraction? Nucleic acid extraction consists of DNA and RNA extraction. It is a routine procedure to isolate and collect nucleic acid, and it is the first step for subsequent molecular, medical, or forensic analysis. DNA and RNA can be extracted from almost any intact human cellular tissue like skin, blood, saliva, hair, mucus, muscle tissue, bone marrow, etc. etc. Now let us have a glimpse about the history of nucleic acids. So following are few of the historical events related with nucleic acids and the major contribution by scientists made in this field. Nucleic acids were first extracted in 1869 by Friedrich Miescher when he was studying the chemical nature of the nuclei of the white blood cells, and he named them nuclein. Next, in 1953, after a long period, after 1869, in 1953, Rosalind Franklin, James Watson, and Francis Crick determined the structure of the DNA, that is deoxyribonucleic acid, because of which James Watson, Francis Crick, and Maurice Wilkins won the Nobel Prize in Medicine in the year 1962. According to their work, it was shown that DNA is made up of two strands, which are long chains of nucleic acid molecules, that is nucleotides, wound around each other in the form of a helix. Later on, in 1968, Dr. Hargobind Khurana, along with Marshall W. Nirenberg and Robert W. Hawley, they won the Nobel Prize for their remarkable work to show the sequence in which the nucleotides are joined together in the strands of RNA, that is ribonucleic acid, and that it can be translated into chains of amino acids to make proteins, which are one of the most important primary building blocks of cells. Now, what is the need of this nucleic acid extraction? The foremost one is we extract nucleic acids, DNA or RNA for our molecular studies. We extract that nucleic acid to study the presence of sequences or amplification of target sequences, that is mutational analysis or cloning, Southern blotting to show the presence of sequences to carry out DNA fingerprinting and cloning of particular gene sequences, sequence analysis for the mutational analysis sequencing of the genome, and for studying the DNA polymorphisms like RFLP, RAPD, SSR, etc., etc. Other application is in the field of medical studies. In that, we mainly focus on understanding the genetic disorders at molecular level and for the rapid detection of genetic disorders in that particular patient. Another important application is in the field of criminology or forensic sciences. This is used for the DNA fingerprinting, for the identification of individuals, and for the paternity testing in any disputed cases. These were the basic application at the School of Biotechnology, RGBV, Bhopal. Basically, in our labs, our research targets type 2 diabetic retinopathy with the help of molecular medicine. So what is diabetic retinopathy? Diabetic retinopathy is a complication of prolonged diabetes that affects your eyes. It is caused by damage to the blood vessels of the light sensitive tissue at the back of the eye known as retina. Prolonged diabetic conditions can lead to the blockage of tiny blood vessels that nourish the retina by cutting off its blood supply. Because of this, the eye tries to grow new blood vessels, or we can say it tries to perform neovascularization, but these new blood vessels don't don't have the capacity to develop properly, and they develop a condition of leakage. As you can see in the figure, a normal retina has having proper blood vessels in it, but in the case of diabetic retinopathy, you can see that it has grown numerous other blood vessels, and which are not developed properly. Because of this leakage, the patient can have severe vision problems like blurred vision, fluctuating vision, impaired color vision, and most important and deadly is the vision loss. Uh, so as to carry out our research in this field, we have collaborated with few hospitals in Bhopal like Hamidia Hospital and Sultania Janana Hospital and few other small hospitals. The need of the collaboration is that we need the blood samples and the retinal tissue samples. So we collect those samples from them, either from the alive patients in the case of blood, or retinal tissues in the case of dead patients. And this is all governed by the ethical committee. So it is all ethical. And using those samples, we isolate nucleic acids and perform genetic and epigenetic studies so as to know the function of the genes related with the progression of diabetic retinopathy, majorly type 2 diabetic retinopathy, and how to build up new therapeutic genes or proteins. So this is our major thrust area. One of our major international collaborator in this field is Wayne State University, USA. We all know that most of the places and most of the institutions in the world do not permit the use of human tissues, but we are able to do so, and this is a plus point for us that we are able to deal with human tissues. And for this, because of this, our research becomes an important uh, it plays an important role in this particular research. So as we know that what are the basic steps of DNA extraction? For the DNA extraction from any blood sample or any other sample, in this basic steps are, first of all, we need to break open the cells and remove the membrane lipids. Then we remove the cellular and histone proteins bound to the DNA by adding a protease, by precipitation with sodium or ammonium acetate, or by using extraction method. Then the DNA or the RNA is precipitated in cold ethanol or, or isopropanol. So this is the basic step in the nucleic acid extraction. So the protocol for the DNA isolation from blood is according to the protocol given by Miller et al. In the year 1998. In this, what we do is we transfer 300 microliters aliquots of whole blood to each of the 3 microfuge tubes. Then we add 900 microliters of 20 millimolar tris chloride pH 7.6 to each tube. We invert and mix. Then we incubate the tubes at room temperature for 10 minutes, occasionally inverting the tubes. Then we centrifuge the tubes at maximum speed for 20 seconds at room temperature. We discard all but 20 microliters of each supernatant. Then we resuspend the pellets of white cells in a small amount of the supernatant left in each tube. Next, we combine the resuspended cell pellets in a single tube and then add 600 microliters of ice cold cell lysis buffer by pipetting several times. Then we add the potassium acetate solution and then we vertex vigorously for near about 20 seconds. Then the protein is precipitated in the form of pellet by performing centrifugation at the maximum speed for 3 minutes at 4 degrees centigrades. Then the supernatant is transferred to a fresh microfuge tube containing 600 microliters of isopropanol. The solution is mixed well, and then the precipitate of the DNA is recovered by centrifuging the tube at maximum speed for 1 minute at room temperature. The supernatant is removed and 600 microliters of 70% ethanol is added to the DNA pellet. The tube is inverted several times and it is centrifuged at the maximum speed for 1 minute at room temperature. We should carefully remove the supernatant and allow the DNA pellet to dry in the air for 15 minutes. Then we redissolve the pellet of DNA in 100 microliters of TE buffer. Then the pellet is solubilized in a time span of 16 hours if it is kept at room temperature, or it can be solubilized within 1 hour if it is placed at 65 degree centigrades. That was about DNA extraction from the blood samples. But as I mentioned, that when we are doing our genetic studies or when we are preparing any therapeutic protein or gene, in that case, our objective is to obtain a gene which does not have introns. As we all know, a gene comprises of introns as well as exons. So in that case, we need to remove any sorts of introns. For that, we need a complementary DNA. What is a complementary DNA? It is a DNA synthesized from messenger RNA, that is mRNA. This reaction is catalyzed with the help of enzymes such as reverse transcriptase and DNA polymerase. Reverse transcriptase helps in the formation of single-stranded DNA from mRNA, and then next, with the help of DNA polymerase, we can prepare double-stranded DNA known as complementary DNA or cDNA. So cDNA or complementary DNA is often used to clone eukaryotic genes in prokaryotes. The speciality, as I said, of cDNA is that it does not contain introns and it just consists of the sequence that is required for our research work. So now comes, not just now we understood that cDNA is prepared from RNA. So what is RNA? RNA is ribonucleic acid and it is a polymer of nucleotides which is made up of ribose sugar, a phosphate and nucleotide bases such as adenine, guanine, cytosine and uracil. Only the difference between DNA and RNA in the case of nitrogenous bases is thymine is replaced by uracil in case of RNA. It is a polymeric molecule essential in various biological roles in coding, decoding, regulation and expression of genes. RNA is a single-stranded helix in comparison to DNA, which is a double-stranded helix. The strand has a 5' end with a phosphate group and a 3' end with a hydroxyl group. It is composed of ribonucleotides which are linked together by 3' to 5' phosphodiester bonds. Mostly, as we just saw, that RNA molecules are single-stranded, but sometimes it happens that complementary base pairing within the RNA strand can happen, and in that case, few regions in that single-stranded RNA we can observe some double-stranded structures because of looping on each other. So it is not a double stranded, it is always a single stranded molecule. So in that case, the RNA can have some double stranded regions. So this is about RNA. Now, what are the types of RNA in our cells? There are majorly three types of RNA present in our cells: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA or mRNA, it accounts for about 5% of the total RNA in the cell. Messenger RNA is responsible for the transcription of the genetic code from DNA into a form that can be read and translated up to prepare amino acids or proteins. Therefore, we can say that mRNA or messenger RNA carries genetic information from the nucleus to the cytoplasm of a cell. Next comes the ribosomal RNA or the rRNA. It is the most abundantly found RNA, and it accounts for near about 80% of the total RNA present in the cell. We all know it is present in the ribosomes, and ribosome consists of two major components: the small ribosomal subunits, which helps in the reading of the RNA, and the large subunits which join amino acids to form a polypeptide chain. This ribosomal RNA helps in directing the translation of mRNA into proteins. Next comes the last one is the transfer RNA or tRNA. This is the smallest of all the three types of the RNA, having just about 75 to 95 nucleotides. It helps in the transfer of amino acids during protein synthesis. This is the reason why they are known as transfer RNAs. Each of the 20 amino acids has a specific tRNA that binds with it and transfers it to the growing polypeptide chain. The following figure depicts the structure of all the three types of RNA. Messenger RNA is a string of all nucleotides and for bases and for sugar and phosphate. Ribosomal RNA, it is present in both the large subunit and the small subunit of the ribosome. And transfer RNA has a hairpin loop-like structure. Once we know about RNA, now we can proceed for our cDNA synthesis. So for this cDNA synthesis, first thing is we need to isolate the RNA. High quality RNA isolation is a crucial step and is required to perform in various molecular biology experiments. The protocol that we follow in our lab is based on the kit provided by Hymedia, known as RNA Express reagent. There are many different types of kits available in the market, but this is the one that we follow. According to this protocol, we homogenize approximately 100 milligrams of tissue in a pre-sterilized and chilled pestle and mortar using liquid nitrogen. The reason for using chilled pestle and mortar is that because we are dealing with frozen tissues, we don't want to raise the temperature and we don't want the degradation of the tissue or any nucleic acid present in that. And that is the reason why we do the homogenization or the sharing in liquid nitrogen. To this homogenized tissue, 1 ml of RNA Express reagent is added and mixed by repeated pipetting. The cell lysate is transferred to a DEPC treated 2 ml Eppendorf tube. So what is the need of DEPC treated solutions or plasticwares or glasswares while RNA isolation is just to get rid of RNases which can degrade our required RNA. The homogenized sample is incubated at room temperatures for 5 minutes. Next, 200 microliters of chloroform is added to each 1 ml of RNA Express reagent. The samples are tightly covered and shaken for 15 seconds, and then they are allowed to settle down at room temperatures for 10 minutes. Then next, the samples are centrifuged at 12,000 RPM for 15 minutes at 4 degrees. The aqueous phase of the after centrifugation contains RNA and it is transferred to a fresh tube, and then 500 microliters of isopropanol is added and the mixture is allowed to stand for 5 to 10 minutes at room temperature. Next, the mixture is centrifuged at 13,000 rpm for 10 minutes at 4 degrees centigrades. After centrifugation, the supernatant is removed and RNA is present in a pellet, which appears like a gel. Then that pellet is washed by adding 1 ml of 75% ethanol by centrifuging at 10,500 rpm for 5 minutes at 4 degree centigrades. After centrifugation, supernatant is removed and pellet is air dried. 300 microliter of free water is added and it is dissolved by pipetting with a micropipette. Once our RNA is isolated from the sample tissue, next we need to purify that RNA. For that, we take 3 molar sodium acetate at the rate of 1/10th volume of RNA sample. Then we add chilled absolute ethanol at the rate of 2.5 volumes of RNA and 3 molar sodium acetate, followed by gentle mixing and incubation on ice for 20 minutes. Next, the RNA is pelleted out by centrifuging at 14,000 rpm for 30 minutes at 4 degrees centigrades. The pellet is then washed with 1 ml of 75% ethanol by resuspension of the pellet, and this is followed by centrifugation at 10,000 rpm at 4 degrees centigrades for 10 minutes. Supernatant is discarded, pellet is air dried and dissolved in appropriate amount of DEPC treated water. Next, we need to convert our RNA into cDNA. For this purpose, we set up our experiment in such a way that we carry out a specialized form of PCR, that is RT-PCR or reverse transcription polymerase chain reaction. As the name suggests, reverse transcription polymerase chain reaction, it it prepares large amount of cDNA using PCR and performing reverse transcription. For this, the following reaction mixture is prepared, in which RNA template is taken between 10 picograms to 1 micrograms. 50 micromolars of oligo-DT are added in the volume of 2 microliters. Then DEPC treated water is added in such a way so as to make the total volume of 8 microliters. Then these reagents which are present in the in that kit is MULB reaction mixture. It is 10 microliters. An enzyme mix is taken 2 microliters. The 20 microliters of reaction mixture is incubated at 42 degrees for 1 hour and the enzyme is inactivated at 80 degrees centigrades for 5 minutes. Then the reaction protocol for cDNA synthesis consists of 4 steps. In step 1, the temperature is maintained at 70 degrees centigrades for a time of 5 minutes. Next step is of 42 degrees centigrades for 60 minutes. Step 3 is at 80 degrees centigrades for 5 minutes. And step 4 is at 4 degrees centigrades, and we can keep this as it is for a longer period of time. Once the cDNA program is completed, then we need to store the RNA at in a required temperature, Now, just now we saw two types of nucleic acid extraction methods. One is DNA extraction and the other is RNA extraction. DNA extraction process deals with isolation and purification of DNA, while process of purifying RNA from samples is RNA extraction. The pH in DNA extraction should be maintained at 8, but in the case of RNA extraction, it should be 4.7. The steps are, first step is the same, that is the cell lysis, but in the DNA, the membrane lipids are removed and the DNA is precipitated. While in the case of RNA extraction, after cell lysis, guanidine thiocyanate or phenol chloroform extraction is done and it is added into isopropanol. DEPC treated water is not required for the preparation of reagents as in case of DNA extraction, but in the case of RNA isolation, DEPC treated water is required. DEPC treated water is used in handling of RNA so as to reduce the risk of RNA being degraded by RNases. The long-term storage of DNA is at minus 20 degrees centigrades, while it is minus 80 degrees centigrades in the case of RNA extraction. Next, we should know about the purity of the isolated nucleic acid. We are actually going to deal with in detail in the next session, but let us have just a glimpse of how we know about whether our extracted or isolated nucleic acid is in its pure form. For this, we use the spectrophotometer. The quantitation of nucleic acids is commonly performed using a spectrophotometer to determine the average concentrations of DNA or RNA present in a mixture as well as their purity. Reactions that use nucleic acids often require particular amounts and purity of optimum performance. For this, we calculate a ratio of 260 nanometers to 280 nanometers. So if the ratio of 260 by 280 is near about is 1.8, then it is accepted pure for the case of DNA. And while in the case of RNA, if we want to check the purity, then the 260 by 280 ratio should be 2. So this is the brief about how we know about how we quantitate our isolated nucleic acid. We need to take some precautions while performing nucleic acid extraction. First, let us know about DNA isolation. Whenever we isolate the DNA, we should make sure that all the plasticware such as vials and microtips that we use, they must be DNA free. Next, we should avoid rapid vertexing of the DNA samples while performing the DNA extraction, so as to just to avoid mechanical shearing and disruption of the DNA. We also make sure that the extracted DNA is treated with RNase to remove presence of any traces of RNA. We can ship the DNA samples on sufficient dry ice. The precautions that we need to take while in case of RNA isolation is that, so as just to avoid any sort of RNAs, we should always wear gloves while handling RNA and make sure that your workbench, pipettes, tips, solutions, reagents, everything is RNase free and reagents are prepared in DEPC treated water. We should use liquid nitrogen to grind the tissues whenever we extract the RNA, just to avoid the degradation of the frozen tissue. We should avoid rapid or frequent freeze and thaw of our RNA, isolated RNA, otherwise it can lead to its degradation. We should always snap freeze RNA samples in liquid nitrogen and keep them at minus 80 degrees centigrades. Likewise, DNA, just like our DNA, we should ship or carry our RNA samples on sufficient amount of dry ice. So this was all about nucleic acid extraction from human. So thank you so much for your patient hearing. I would just like to sum up this session of DNA extraction or nucleic acid extraction from animal and human beings. So the summary of this session is, it was actually divided into two. in two parts, that is DNA extraction and RNA extraction. So the basic steps of DNA extraction are: the cell and the nucleus are broken open to release the DNA inside using detergents or enzymes such as proteinase K to free the DNA and dissolve cellular proteins. Alcohol such as ethanol or isopropanol is added that leads the precipitation of DNA, and rinsing of the DNA pellet with water or tea buffer or any other buffer. Next is the basic steps of RNA extraction. For this, the tissue is homogenized in the chill mortar and pestle in the presence of liquid nitrogen, then performing chloroform extraction, washing of the RNA pellet with 70% ethanol, and resuspension of the RNA pellet in DEPC treated water. So now I would like to invite Miss Shivangi Srivastava. She is pursuing her MTech Biotechnology from School of Biotechnology, RGPV Bhopal. Before her presentation, I would invite any sort of queries related to the first session. So the session is open for any queries. And later on, Ms. Shivangi Srivastava will continue with her presentation.
Okay. Thank you, Riddhi. There are some queries in the chat box. So,
Okay.
so I am entertaining one by one. The first
Okay.
is how does DNA recovered from crime scenes? It by the question. Okay.
Okay.
So I would like to say there was a scientist, namely Locard. He has given Locard exchange principle, and as per this principle, he said that if any crime scene taken place, the criminal have to replace their things, maybe in the form of skin, hair, or some tissues, blood. Means he always takes something from there with him or with her, or he left something there at the crime scene. So with the help of blood stain, nails, some tissues within the nails, maybe, so we can use these particular samples for the isolation of DNA or recovery of DNA for the identification of criminals or the crime scene. And
Yes.
second question is why DPC treated water used? Okay,
Yeah.
so DPC is nothing. DPC is diethyl pyrocarbonate.
Yes.
Okay, so by treating this, they we are going to inactivate the RNAs that in the water. Okay, it is only used by to inactivate the activity of RNAs. So it will minimize the chances of rupture of RNA while you are going for nucleic acids isolation. So
Exactly.
while you are doing all such type of works in your laboratory, while you are handling RNA, DNA or isolating nucleic acid, you always use DPC treated water. And another query is that is somebody have asked ki, which was the method, which is better method to use for isolation of metagenomic DNA? It was the question. So the isolation of metagenomic DNA, the method already or always could be decided on the basis of yield of DNA, how much DNA you want to isolate, quality of DNA, and further applications, or we can say that the downstream processing, what you will do with that particular DNA. So there are basically two methods. One is manual, another one is kit.
Yeah.
So there are so many kits commercially available in the market. You can buy it and you can use that kit for the isolation of DNA, metagenomic DNA, by their respective protocols, by following
Yes.
protocol. But If you are doing such type of work, we prefer always manual methods, because there are chances to manipulate the method as per your requirement of yield of DNA. There are chances to manipulate some protocol method just to increase the quality of DNA, and it is useful for your further applications. And another one is that query there is, that is why uracil is replaced by thymine in RNA. It was also a query. So I would like to say the basic difference between DNA and RNA, always remember, is the presence of sugar. In DNA, there is deoxyribose sugar, whereas in RNA, there is ribose sugar. Okay, so the uracil replaced thymine while transcribing mRNA or DNA to mRNA. It comes uracil in place of thymine. It is due because always form uridylic acid, while this is combining with the base and ribose sugar. We can say nucleoside, not nucleotide. So it always comes uracil rather than the thymine. Thank you very much.
Thank you, Dr. Suresh. How temperature is set in the centrifuge? It is available in that machine itself where you can set the required temperature.
Then okay, now we can go for another session.
Yeah, I think so.
Okay.
Ms. Shivangi, please continue with your presentation.
Okay, thank you so much, Reddy ma'am and Suresh sir, for elaborating the basic knowledge of DNA, RNA, biochemistry of reagents, with history and how to isolate the DNA. So I'm here to describe you all about how to extract the DNA from plant cell. So the basic of plants are, like, plants are living organisms and is a part of Whittaker's five kingdom classification of organisms. And plants are eukaryotic multicellular autotrophs. What is the meaning of autotroph? Actually, autotrophs are those which produces its own food with the help of sunlight, soil and water, and this process of producing their food is known as photosynthesis. They are non-motile, as we all know. They can't go anywhere. And what is a eukaryote? A eukaryote is an organism whose cells contain complex structure enclosed within the membrane. And eukaryotic cells have organelles and organized DNA. As we all know, our basic as we all know, all the characters are stimulated by the genes. And then genes, what is genes? Genes are the small segments that is present on DNA. Okay, so DNA can be help for showing any character, like it will show the character of producing food or organelle membrane and everything that is enclosed within the membrane. Okay, so there are organelles like cell wall, that is polysaccharides present in the matrix of lignin. Cell membrane, it is a phospholipid, it is made up of phospholipids. Cytoplasm, in which all the organelles are present. And mitochondria and chloroplast are the powerhouse of a cell, and chloroplast contain n number of colors, pigments, that will show on that time. Central vacuoles contains water in the cell. Lysosomes contain digestive enzymes. Golgi, what is rough endoplasmic reticulum, ribosomes, nucleus and nucleolus. Our DNA is present in the nucleus and in the nucleolus. So we have to extract the DNA from there. As we all know, many times in our Viva, generally question arises that what is the difference of isolation and extraction. So most of us often assume that both extraction and isolation are same, but there is a difference. There is a key difference between them, is that extraction is a technique that helps to separate the desired compound from a mixture that is a crude form. As we are going to talk about the DNA isolation, that means the extraction of DNA which contains the impurities of RNA as well as protein. So this is the process is known as extraction. But the isolation is the technique that helps to purify the extracted compound. As we all know, as we want to isolate the DNA, so isolation is the process which removes all the impurities of protein as well as RNA to purify the DNA. So why isolation of DNA is required? Because it is very useful for the study of genetics, biochemistry, ecology, evolutionary biology of organism. Some of the examples are here. So it is used for determination of evolutionary relationship of a plant with its ancestors. Okay. So to know the origin of certain plant species, used in molecular biology to know the functions of gene like mutations. As maybe some of you watched the TV in which an advertisement of sugar free showing a genetically modified sweetener producing plant, that is tulsi. In English, tulsi, in botanical name, tulsi is Ocimum teniflorum. So they genetically modified that tulsi and produces lactose from them, and now the tablets of sugar free of that lactose are under mass production. Okay. Used in recombinant DNA technology, like for producing vectors and hybridization experiments of DNA. And last one is the for the mass production and analysis of novel biological products. So before experimentation, we have to take some steps that are set water bath at 65 degrees centigrade and incubate 2x CTAV solution prior to use. We are using CTAV method for isolating plant. So 2x CTAV solution prior to use incubated. Okay. So add beta mercaptoethanol to CTAV buffer right before the use and make 70% ethanol by mixing distilled water in 95% ethanol. So how to isolate the DNA from plant cell? Here are some steps which are showing the isolation of DNA from the plant leaves. So collect the fresh leaves, because fresh leaf contains freshly synthesized DNA. So it is easy for any type of study, and wash properly and soaked with uh, the help of tissue paper, soaked in water with the help of tissue paper. Okay. Remove the veins of leaves, weighing properly, and soak in absolute alcohol for 1 hour. After that, dry them with tissue paper. Slowly preheat CTAV buffer at water bath of temperature 65 degrees centigrade, and add in sample leaves. Then grind them with the use of mortar and pestle. After that, collect the slurry in a pinch of tubes and add beta mercaptoethanol in tubes, and then go for the incubation for 1 hour at 65 degrees centigrade. After that, add chloroform and isoamyl alcohol in the ratio of 24:1 and gently mix it with inverting. After that, going for the centrifugation. Centrifugation is done at room temperature for 10 minutes at 10,000 rpm and collect the supernatant. Why? Because the reagent which are used for precipitating the RNA or protein contaminants or for cell debris. Okay, so that cell debris come in the palate. So for our interest, for our product, supernatant is used. DNA will be present in the supernatant, not in the palate. Now add equal amount of chilled isopropanol, mix gently, and incubate at -20 degrees centigrade for 60 minutes or for overnight. Okay. Now again centrifuge. Before this step, when you take your DNA or your sample from -20 degrees centigrade, do not throw them, because if you are throwing that sample, then that will be the more chances for degrading your DNA structure. So you can't get any result from that. So don't thaw your sample from when it comes in normal temperature, then it will go for further centrifugation and centrifuge at 10,000 rpm for 10 minutes and wash the pellets. In this, pellets are useful because our DNA is present in the pellet, and all the impurities present in the supernatant that will be discarded, okay, with 70% ethanol. Now again centrifuge again at 10,000 rpm and for 5 minutes and discard the supernatant. As I told you that supernatant contains impurities. With drying the pellet and add 0.1 x t buffer and incubate at 37 degrees centigrade for 1 hour. After that, store at minus 20 degrees centigrade for further use or for further study or for the quantification, analysis, etc., etc., so on. After this, we have to know the uses of chemicals. Why we are using that chemicals, that reagents, or that buffers. So here are some examples for showing the biochemistry of that chemicals. So first of all, why we use CTAB buffer? Because CTAB is a cetyl trimethyl ammonium bromide. That is a cationic detergent and that has useful property of precipitating nucleic acids. The CTAB buffer, extraction buffer, is used to maintain an environment in which the DNA will not be degraded and help to separate the DNA from other cell components. As we all know, DNA have negative charge on it. So this detergent is in cationic nature. When cationic detergent is come in contact with the negative DNA, it will form an ionic bond. Okay, so it will precipitate the nucleic acid and remove all the impurities. After that, vortex with phenol is often effective for breaking down proteinaceous cell walls. The addition of a detergent such as SGS is often necessary to remove lipid membranes. The extraction buffer is used to maintain an environment in which the DNA will not be degraded and help to separate the DNA from other cell components. Here are some also, like Chris, as someone asked in their query, say in chat box, what is TE and what is the role of TE? Here is the answer. Tris maintains a constant pH and EDTA chelates the metal ions. It chelates Mg2 plus, which is essential cofactor for the DNase enzyme, which is and Mg2 plus ion have important role in maintaining the sorry in maintaining the phospholipid membrane. Okay, so EDTA chelates that metal ion and allow all the organelles come out from the membrane. Beta mercaptoethanol is a reducing agent which protects the DNA against quinotes, disulfide peroxide, and polyphenol oxides. And we also know the property of beta mercaptoethanol. It is very useful for degrading the protein. It degrades the disulfide bond which is present in the protein that helps to degrade the protein structure. Okay. So NaCl provides sodium ions that will block negative charge from the phosphates of DNA. As we all know, DNA have phosphate group, and that phosphate group showing the negative charge on the DNA. So the negative charges on DNA cause molecule to repel each other, and it disrupts the DNA structure. The sodium ions will form an ionic bond with the negatively charged phosphates on the DNA and neutralizing the charge and allow them to come close. Here are now everything is going right. So here are some precautions which we have to be taken while experimenting and while isolating the DNA from plant leaf. The working platform must be clean before performing the experiment. Otherwise, contamination will occur and your DNA, you didn't get any DNA. The pH of all the buffers and reagents should be maintained before the use. Pipetting should not be done from mouth. Preparation of volatile compounds should be done in fume chamber. Why? Because some volatile compounds are hazardous to our health, so it should be taken like a precaution. This work is done in only fume chamber. Do not use plastic containers while working with phenol and chloroform, because in the higher concentration they can able to burn the plastic containers. And proper discarder should be used for disposal, because we all know the hazardous properties of all the chemicals. So if you discard it as like in a land, then it will cause some severe condition for your health and for many organisms health, not only for humans. It comes for all type of organisms. Wear PPE kit while performing experiments. Glassware should be autoclaved. So now here I am going to summarize this session. In this session, what we have discussed. We discussed in short of in brief description of about plant and plant organelles, and what is the difference between extraction and isolation, and what is the need for DNA isolation from plants, and DNA isolation technique, how to we isolate the DNA from plant cells, and the study related to the reagents biochemistry. Now, I would like to invite Dr. Archana Tiwari, who is Faculty of Applied Science in the RGPV and Director of our School of Biotechnology, RGPV, for summarizing for today's both of the session.
Thank you, Ms. Shivangi Shrivastava, for nice presentation.
Thank you, ma'am.
Now her presentation is open for the discussion. If anybody is having any queries, kindly put that queries so that we can try to resolve your queries. First, we will go for this, then we will conclude today's both the sessions. Any query by anybody else?
How do you deal with the DNA contaminations, like viral DNAs?
Anybody is asking about the DNA contamination?
Yes, ma'am. Yes.
Sure. What is your question? Kindly repeat your question.
It's if we are extracting the plant DNA, but sometimes the plant is infected by the virus.
Not audible,
Then
please.
that,
You're asking about DNA contamination.
yes, ma'am.
Yes. Right. Shivangi, he's asking about the plant DNA contamination. Am I right?
Plant DNA contamination? Which type of plant DNA? Every when you are going to isolate any DNA, there will be contaminants of RNA and protein. So you should check on the basis of quantification, and in quantification you can check by spectrophotometer by various reading on the ratio of 260 to 280 nm.
Very good.
So you know the contamination from there.
Tomorrow we have one separate session that is for the purification and quantification of the pure DNA, whether whatever the DNA is extracted from either from animal, from humans, from plant, from viruses, from microbe, that is bacteria, whether that are in the purified form or not. If they are in the purified form, whether they are in the ratio of 260 to 280 or not, whether they are having the 1.8 or 2 or not. If they are not, so they are considered under the category of contaminated DNA. If they are, so they are considered under the category of pure DNA. We will deal tomorrow also, right? I think your query is satisfied. Anybody?
I was
Yes, please.
asking about that. If it's other means undesirous DNA, this has been contaminated in our desirous DNA.
If the contamination occurs in your desirous DNA, am I right?
Yes, yes. Of viral DNA, like viral DNA. If a plant is infected by the virus and somehow get the DNA extracted from the plant, but you get the viral DNA. So how did you get that differentiation that it is a viral DNA and it is a plant DNA?
Whenever you're designing your experiment, we have to take care of all these. Whether we are dealing with the plant which is infected with the virus, or it is free from the viral infection or viral infection or any of the infections, right? Whenever we are designing our experiments, we have to just focus on all these points. For instance, we are dealing with the cell lines. Mycoplasma is present in the cell lines, that is in the cell tissue. When we are dealing with the cell lines, we are adding the antibiotics, no? So just to separate the mycoplasma from the desired cell lines. In the same way, whenever we are designing our experiments related with the plant for the isolation of the DNA, what we have to do, we have to take care of all these things. Then only we can differentiate that this is the DNA which is interfering during the separation of plant DNA, animal DNA, or any of the DNAs. Am I right?
Yes, ma'am.
Sure. Anybody else is having any query? Riddhi, we please go through the chat box. Somebody is DPC water. Yes.
Yes, ma'am. Yes, you can certainly store your DNA in DPC treated water, but it is more advisable to be used to store RNA because it helps in RNA's removal. So that is more advisable.
For the DNA, there is a need of T buffer. That is more than
Yeah,
sufficient.
more than
Right?
sufficient. Exactly.
And DPC treated, that is RNA is very, very delicate fragment. It is degraded very quickly, very fastly. That's why there is a need of DPC water. Am I right?
Yes, ma'am.
Anybody else wants to ask anything? Anybody else is having any query? Shall we proceed to just wind up both the sessions? Now, if there is no queries, we are going to wind up our both the sessions. Today, we dealt with the two sessions under which we have discussed about the various aspects of how to extract and isolate the DNA from animal tissues, taking the example of blood, and in the same manner, extraction and isolation of DNA and RNA from human tissues. One example was taken by Riddhi Tiwari as blood, and another example, whatever she is doing for her doctoral work, that is the isolation of RNA from retinal, that is donated retinal tissues, which we are collected from the hospitals of Bhopal, which she has mentioned in a presentation also. Then in the second session, we dealt with extraction and isolation of DNA from plant tissues. We hope these two sessions are beneficial for you. If you are having, if you are having any queries, kindly just put that queries in the chat box so that we can try to satisfy that queries. Now, tomorrow at the same time, on the same platform, we will start our sessions with the extraction and isolation of DNA from RNA from viruses and extraction and isolation of DNA from bacteria. Followed by the another session, that is purification and quantification of DNA and RNA. N number of participants are having various queries regarding the purification and quantification, right? We think tomorrow your queries will be satisfied. The purification and quantification of the DNA and RNA, followed by gel electrophoresis of DNA and RNA, under which we will deal with preparation of the gel, loading of the samples, running of the gel, and visualization of the DNA bands on the gel, followed by the feedback session. As we have already mentioned in the mail, whenever we are sending you the URL, please stick with the time. Still, the people are joining till the end of the session. Today we have permitted, but it is our humble submission. From tomorrow, we are not going to permit any of the participants after 15 minutes of beginning of the session, because it creates lot of hindrances to the presenter. I'm just underlining inverted commas. Please careful about the timings. If you are not at all having the time for the such type of workshop, absolutely no problem. But please do not create hindrances during the presentation. Now, the time to take as much as vitamin P you want to take till the tomorrow's session. As we know very well that P stands for positivity, and n number of things are demarcated in the P, which clearly states when we are taking such type of things, definitely the positivity will come. Stay safe, stay khush. We will meet tomorrow. Better days are ahead on the same platform at the same time. Thank you so much. Till then, goodbye. We are just ending the meeting here and stopping the sharing of the presentation also. Thanks a lot.